Hi, welcome back. So far, we've spent a lot of time discussing the quantification in cell biology of spatial scales, meaning sizes. In this context, we went over areas, volumes, mass, densities, and other properties that can be considered to be the spatial aspect. But now we're going to consider time. And I'm going to try and make the case, as I hope you will see, that time and energy are interconnected. This is sort of a following section, and this will take a little bit of effort, but I hope you will see the connection between the segments. Now, if I say energy to a biologist, they always instantly will say ATP is the energy currency of life. Yes, and we're looking at an ATP molecule, adenosine triphosphate. Um, those of you who remember their DNA biochemistry also remember and realize, perhaps looking at this, that the ribose sugar has its OHs intact. There's a bond to an N group that comes from H and an O phosphate, 1, 2, 3, triphosphate. Therefore, adenosine. So this is the adenine, osine because of the ribose, triphosphate. Okay. This is alpha, this is beta, this is gamma phosphate. The chemical reaction that we are usually interested in considering with regard to ATP in its role of energy donor is ATP in the presence of water loses a phosphate Pi at the cost of release of 7.3 kilocalories per mole of energy. Now, how this affects time scales, we'll come to. What you're looking at here are bacteria moving in a movie that is taken from Howard Berg's lab. Howard Berg is a pioneer in bacterial biophysics. He developed microscopy methods to image single E. coli cells. Again, E. coli was chosen because physicists considered that to be a model, ideal, simple cell. And the flagella are labeled by Alexa. The whole microscopy was done in fluorescence. What you should see is that these flagella vibrate vigorously and move and propel the cell forward. In contrast, a movie of a single neutrophil demonstrates cell migration at its most apt. This movie is taken by David Rogers at Vanderbilt University, and it involved adding Staphylococcus aureus bacteria, which is this very small circle here, and you see it here, 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 as it is being chased by a neutrophil. This is taken from whole blood, human whole blood, and the neutrophil moves as it senses the direction of the bacterium, turns, and after some time actually captures it. But we are interested in the rate, the time scale, right? So we are interested in knowing how fast a cell migrates. So in order to address that, if we use direct observation as a method, then using the scale bar of 20 microns, knowing that it moved in 1 second by about 1/20th of that, we know it is moving 1 micron per second or 60 microns per minute. And if we are to compare it to anything real world, then we can consider how much it moves per meter. And I want you to do the arithmetic, and we will discuss this in class, how fast does a cell migrate in meters per second. Okay. So the speed of cell crawling is approximately a micron per second. The speed of bacterial motion is about 20 microns per second. And we can in fact put all the time scales, the times taken by cellular systems to move onto one single scale. Just as a reference, I have pointed out that human lifespan is, if we are healthy and live to our ripe age of 70, then 10 to the power 9 seconds. This is what we call order of magnitude scale, where we have used the 10 to the power something values to depict tenfold jumps in seconds. So 10 to the power minus 9, I'm sorry, thousandfold jumps in time. 10 to the power minus 9, minus 6, minus 3, 10 to the power 0, plus 3, plus 6, plus 9, plus 12, plus 15, plus 18. All this in seconds. Plus 18 at the upper limit is maximal biological time scale of Earth, 10 to the power 17 seconds. As far as we know, there is no life on any other planet other than Earth until we find it. We're looking. There are many exciting projects on extraterrestrial life and search for extraterrestrial life. Diversifications of humans and chimpanzees happened 6 million years ago. That's 2 to the 10 to the power 14. Sequoia is a giant tree found in Western California in Northern America. It's 10 to the power 11. Galapagos tortoise has 10 to the power 9. Human embryonic stem cell has doubling time of 10 to the power 5 seconds. E. coli, which we talked about earlier also, has a doubling time or takes about 10 to the power 3, 1,000, few thousand seconds to divide. Unstable protein half-life is 5 minutes, meaning to say the time it takes a protein to degrade is about 10 to the power 2 seconds. That is 5 minutes. Lysosomes turnover rate is 0.5 per second. And then we go faster and faster and faster, with the fastest speeds being smallest times being covalent bond vibrations in water of 1 femtosecond 10 to the power minus 15. So knowing this, at the cellular scale, the time scales we're interested in are typically in the range of 10 to the power 3. In order to estimate, to measure the time it takes a process, similar to this question that we asked earlier about measuring the number senses, we need techniques. And one of the techniques is called fixed time point analysis. I showed you an example with the cell migration. The second is direct observation, and third is pulse chase. These are the 3 typical methods that have been used vastly across biology to quantify. Remember, estimate time scales means actually measure time in seconds, not arbitrary units. And I will get to this again about the difference between units, dimensions, and the need for precise physical units in quantitative biology and biophysics. So fixed time point analysis is based on the idea that you can look at a stage in, let us say, the development of an embryo or of a Drosophila fly, from the stage of when it is born as an embryo to the first instar larva, to the second instar larva, third instar larva, which is this creeping worm-like object, to the pupa where it stops moving, to the male adult. Knowing the stages, identifying them, measuring them hundreds of times, now gives us a time scale or value for how long it takes to go from embryo to first instar, etc., etc., etc. Incidentally, most of the work on establishing Drosophila as a model organism was done by Thomas Hunt Morgan in 1910. And he selected for rapid generation time ease of breeding and clear delineation of times and stages, meaning to say he could identify the specific stages and the times they took to go from one stage to another clearly. Early development is a little more involved, and a lot of developmental biology and embryology involves identifying the exact stages of early development. Most of these images, by the way, are taken from single time points, and then another fly is imaged after 5-6 more hours, and another fly is imaged after 20 hours, etc., etc. Which is what the idea of fixed time point analysis is. The same individual is not observed throughout. It is from this that we know that the process of fertilized egg embryo, one staged embryo, to 9 nuclear divisions, to nuclear peripheral migration, to cellularization, and then the entire development at 10 hours is critical for the growth and development of a fruit fly. Another classic fixed time point analysis is bacterial growth curves. What you're looking at here is the translated cells per ml number for a growing bacterial culture. So you will in fact find that the measurement is based on something that some of you may have heard of earlier, optical density at 600 nanometers. But optical density per se does not have the units of cell density, cells per ml. So how do we convert? We need a conversion factor. Typically, for E. coli, we consider 1 OD to be 10 to the power 9 cells per ml. This is a conversion. For every bacterium, this has to be different and needs to be checked before you can use something like this. This data has points which each individual point is sampled by taking a flask in which bacteria are growing, removing an aliquot, taking out some amount of the culture, taking it to the spectrometer, and measuring the absorbance, and so on and so forth for all the time points. And this is how we get our growth curve. If we want to estimate general properties like growth rate, we fit it to this equation, which is a logistic equation, and allow and use the standard definition of growth rate to identify the doubling time, which is the ratio of the natural log of 2 to r. So a second method of measuring time scales is direct observation. Cells are micron scale, 10 to the power minus 6 and larger. So we need a microscope because the human eye has a limit of seeing objects. This is approximately around 0.1 millimeter, 100 microns. So anything smaller than that, you need a microscope. You, in fact, cannot see structure at 0.1 mm. You just see a dot, tiny dot. So most of these dots I'm making are around a millimeter. If I make anything smaller than that, you will not be able to see it. Optical microscopy is a very powerful tool and came out of the work of Leeuwenhoek. And a whole series of biologists, inventors, physicists, opticians who went on to improve the methods. In combination with the biochemistry that allowed us to get molecular tags for antibodies or genetically encoded markers like GFP, microscopy has become almost the most favorite method of all cell biologists because it also allows you to directly observe. So for example, I can directly observe a bacterium growing and measure the time scale of its growth because I can see from time t0 to time t50 how long it took for the, which is what I find typically in these experiments, that it took the cell to grow. This is taken from one of our papers and you can find more details in the manuscript. The idea is, in other words, to do microscopy while watching so that you catch the cell in the act and mark time. But there's a more traditional approach called pulse chase. For the purpose of measuring translation time, which is thought to happen in roughly half a second, one protein translates in about half a second, yes, we need to follow the ribosome movement. For that, the polypeptide synthesis needs to be followed, and for bacteria, it turns out this number is about 10 to 20 amino acids per second being added. For eukaryotes, it's 3 to 8 amino acids per second being added, which would mean that about 100 to 200 amino acids would be added. Amino acids per second is 10 to 20, which means about 10 seconds are required for a full protein synthesis, at least. Pulse chase involves feeding the cells with a radioactive analog of an amino acid, which has 32 microcuries of 35S, that is radioactive isotope of methionine, in the methionine integrated in it. 6 to 8 seconds later, the protein is translated. 1 ml of samples are taken ice cold every few minutes, and 2D gel analysis used to know the time course. Now, in this particular case, you also need to detect radioactivity because it's only the labeled protein that you're interested in. So this part, methionine, labeled, is followed by a chase of unlabeled methionine. And this pulse and chase means that this methionine goes into the amino acids, increasing, and because it is chased, it will reduce. And knowing the rate, that is to say the rate of increase of radioactivity per unit time, the slope, we get the translation rate. This is work described in 1984 by Peterson. This is classic method of pulse chase. But for second and sub-second processes, things get a bit complicated. And ion channel gating, for example, is in picoseconds. In milliseconds. So in order to do that, you need faster measurements. In the case of ion channel gating, patch clamp method was invented as a method to try to catch the opening and closing of ion channels. And this is a very powerful method, again, awarded Nobel Prizes for its invention. Biochemical reaction timescales can be even faster, 10 to the power minus 6 seconds, and you need stop flow methods, microsecond freeze, hyperquenching, and other approaches. So to summarize, direct observation, fixed time point, and pulse chase, and product accumulation are all methods to get timescales, but they depend on the scales that you're observing. Just to give you an example, the kind of outputs that we get tell us that chemotaxis, random walk, and flagellar rotation are at different timescales themselves. The speed of directed motion is about 30 microns per second. Flagellar rotation is 100 units per second, or rotations per second, which means 10 milliseconds is the time for one rotation. And the dynamics of the proteins are at an even faster time scale. So scale of size determines the scale of time. Things that are small do things faster, things that are big do things slower. So in a way, we would like to, when we compare time scales, you remember I mentioned about how fast a bacterium or fibroblast moves, or neutrophil moves. You can, of course, take the fact that it moves at about 1 micron per second and say 10 to the power minus 6 meters per second. But you have to also perhaps consider that the time scale is dependent on the size. So if you now take the world record of 50-meter swimming of 20.91 seconds for a human being, how can we compare this? Can we even compare it? Can we ask and answer the question of bacteria relatively faster than humans? So we say that if human swimming is 50 meters performed in 20.91 seconds, it's 100 meters in 40 something seconds, so 41.8. We account for body size. It's about a meter. Human body size is meter. Scale is the distance traveled per second by body length, meaning to say how many body lengths does it cover. So if it covers 50 meters in 20.91 seconds, how many bodies has it covered? Assuming that the human body is about 2 meters in length, which is a bit of an overestimate, not everyone is 2 meters, but Olympic swimmers can sometimes be very tall, then the human is doing 1 body length per second. Bacterium, on the other hand, is covering 30 microns per second, which is 10 times, actually exactly 15 times the body length in 1 second. In that sense, you could argue that the bacterium is swimming faster. Yeah? And a similar set of analogies could be made for human running at 100 meters athletics, which is 9.58 seconds. I think that record is held by Usain Bolt, but I urge you to check your general knowledge. Another process in which time plays a very important role is the cell cycle. In fact, it's a cycle because it's clock-like. And it's so important that the discovery of the molecules involved in it were awarded a Nobel Prize for Physiology and Medicine in 2001 to Lee Hartwell for cell cycle start and checkpoint, Saccharomyces cerevisiae, and Paul Nurse and a third person. The Tim Hunt for the cyclin discovery. Cyclin discovery was in a not very typical organism called the sea urchin ardachia. And Paul Nurse discovered cyclin-dependent kinase in Schizosaccharomyces bombi, not the brewer's yeast, but a yeast found in East Africa in locally made beer. Now, what cell cycle really is stunning in terms of its synchronicity is observed in a xenopus egg development, where synchronous division appears to almost follow a clock-like precision. The genome, by the way, is 10 to the power 9 base, with 3.1 into 10 to the power 9 base pairs of xenopus. It's distributed over chromosomes, but the doubling time is only 30 minutes. This is amazing because you have to divide a billion bases, more than a billion bases, in 30 minutes. Replicate, divide, segregate, and so on. The precision with which this happens is in itself a fascinating question and not one of the very well understood problems. When we look at embryonic development itself, the time scale of development is determined by the doubling time. And that tells us that if there are two cells, if there's one cell, then we have 2 to the power n cells for n number of generations. And the generations are given by the time divided by the cell cycle time. Xenopus laevis, whose embryos we saw earlier, are a classic model organism, both for embryology as well as for developmental biology and cell biology and cell division research. The females of Xenopus are much bigger than the males. This is unlike the human height data, which shows men to be bigger than females. Human males are bigger than human females. The biochemistry of Xenopus oocytes is such that when you harvest them in unfertilized stage, their meiosis is unarrested. So you can make a huge cytoplasmic volume with them of a few milliliters and use that for biochemistry. And it was in this that it was demonstrated that the transition from interphase to mitosis is accompanied by an increase and drop in cyclin associated also with the H1 kinase activity. This was seen using Western blotting and kinetics. This led to the model that the cell division is triggered by an increase in mass, and this mass is being monitored by the proteins that are driving this increase, and that there is a concomitant increase in DNA replication, which then leads to splitting at the time of division, segregation, or anaphase, you may say. And this led to the discovery of the G1S, G2M famous stages of mitosis. So in a sense, the timing of the cell cycle would be chaotic were it not for the fact that there are multiple checkpoints. And these checkpoints, for example, from M to G1 phase, ensure that the chromosomes are attached, otherwise you will have aneuploidy. Check that centrosome duplication happens. Ensure there's no DNA damage. And there is no overduplicated DNA already present before division. So the timing of the cell cycle is crucially not just regulated by the clock that drives it, but also by the checkpoints. And this has led many people to speculate what mathematical model or biophysical model can we construct in terms of equations for the model of the cell cycle. So something that goes up and comes down, goes up and comes down, suggests that the cyclin rise and its threshold dependent triggering of cyclin dependent kinase CDK, which comes secondary. So cyclin rises and later on leads to a spike in CDK, which in turn degrades cyclin, resetting the clock, can be modeled. But the strange part is that the geneticists have found a lot of factors involved, and this is all found by knockouts. They found that this pathway, while very complex, is robust and involves multiple interregulatory inputs. It always begs the question, why is it so complex? But I don't think we can answer this why question. I don't know, and the field is not very clear. So largely, the consensus is that it is likely to be a part of the robustness. Complexity is a part of the robustness. So the budding yeast cycle looks like this beautiful image from Haley's lab at the Yeast Research Center, showing G1S, G2M transition. But not just yeasts show cell cycles, even bacteria like the Colobacter crescentus in a DNA microarray show that there is a timing of gene expression of certain genes, early, intermediate, and late, whose progression determines the cell cycle. So almost like eukaryotes, Colobacter crescentus, a bacterium prokaryote, has a G1, S, and G2 stage. This stage involves going from swarmer to stalked to pre-divisional to then dividing, and corresponds with DNA replication and chromosome segregation. E. coli, on the other hand, our standard model, has a slightly more complicated replication mechanism. So while on the one hand, replication can proceed in the semi-conservative fashion, that the polymerase unwinds, the helicase unwinds the DNA, the polymerase in the forward direction goes forward, the Okazaki fragments are made, and you get semi-conservative replication. The DNA is made, the RNA polymerase transcribes, and then translation happens. This is nothing but the central dogma that you're looking at. Of molecular biology. So DNA polymerization rates are about 252,000 base pairs per second. That's very high, by the way. But E. coli can grow and divide in 1,000 seconds, which means that it takes more time for the DNA to replicate than it takes to divide. How can that happen? Turns out that E. coli fires more than one replication fork and in advance predicts its fate in the next generation, and therefore takes care of the lag. One more example of timekeeping mechanisms or clock mechanisms is the most obvious. Most of you, unless you are now working in a call center, working for United States companies, wake up with the sun and go to sleep at night after you maybe had some discussions with your friends or meeting them and hanging out. If you're lucky, if you're a farmer, you have to wake up with the sun and go to sleep with the sun because the next morning is very hard. This day-night cycle is conserved across most animals. In fact, it looks like even single-celled organisms have day-night cycles. And it's endogenous. And it's amazing because it's coupling something that is planetary, that is movement of the Earth around its own axis, pointing away from the sun and slowly, gradually moving away towards it again. And it is seen in this actogram, that is an activity monitor of Drosophila movement, showing that circadian rhythms of flies are modified in mutants for per and clock genes. These per and clock genes were identified by these groups, Panda and company, to be essential for the process of circadian rhythm. But in order to find a simpler organism in which this happens, people searched and found a blue-green alga called Cynococcus elongatus, in which a minimal circadian oscillator, chi A and B and C, function, and even in vitro will give you a beautiful clock-like pattern over 24 hours. That is, if you notice here, NP chi C is dark, becomes light, and returns to dark, all in the space of approximately 24 hours. So one cycle is completed in 24 hours. This is reaction done at 30 degrees Celsius. There are temperature-dependent processes that affect the dynamics of the Cynococcus circadian cycle, but there's no degradation involved. It's only phosphorylation. So in order to understand something, we try to model it. We try to make a mathematical model, an equation, and try to compare it to reality. In the case of cell cycles, the minimal oscillator, cell cycles and circadian rhythms, the minimal oscillator description is this. There is an activator, that is A. There is a repressor, that is R. Activator activates the repressor and itself self-activates, and repressor inhibits the activator. This is a feedback loop, a negative feedback loop, and auto activation. This can be used to describe either the embryonic cell cycle oscillator or the circadian oscillator. This is really surprising because these are very different kind of phenomena. But the timescales of biological clocks can be described by these simple feedback models fairly well. Of course, that raises the second question which I had asked earlier, which I think is hard to answer and has not been answered, which is why does the complexity exist? We say robustness, we don't know exactly in the case of each of the systems what it is. Indeed, there is a diversity of circadian rhythms from cyanobacteria to birds to animals to mammals to Drosophila to Neurospora, and the time and the nature of the cycles and the mechanisms are different, but they all seem to have a positive component, a negative component, and either degradation or phosphorylation, and there is a connection with clock genes. So it would seem that timescales in cellular life are set by very similar conceptual maps. For the last part, I'm going to talk about energy scales.